disseminate the advancement happening around circling this pathogen. Professor, you are more than welcome. All our faculty members, they are all ears to you, and we are very much happy to hear to you. Please proceed.
Right, thank you. Good morning to everybody, to all the participants, and also for the organization of the meeting. I'm, as I said already, Francesco Pomiglio. I am a vet. I work for IZASAM, that is part of
wildlife लगता है उसमें भी chances है zoom में लगने के वहाँ पे तो जानवर होते हैं
Rabia, please, you need to close the microphone. Sorry.
Wildlife है, agriculture तो है, wildlife भी है, फिर अब्बू सही.
Okay. Maybe it's better to ask Rabia to close the microphone because it's impossible for the others. Okay, thank you. So I am the leader of the unit of food hygiene here at Istituto. And as you can see at the website of Istituto, we are in charge of some national reference laboratories and also collaborating center for OIE and WHO. So the idea for today is to share three aspects of the work that we are doing here in Istituto, and through three different presentations on Listeria, because I am particularly involved in the work of National Reference Center, National Reference Lab for Listeria monocytogenes, and this is one of the most important interests in the part of research and development. We are not a lab of research in the strict sense, because we do mostly research application in the different food of interest, but we are developing some new competencies and tools that will be we will be able to share with all of you if you are interested, and starting from the university and starting collaboration in the framework of OIE Collaborating Center for Food Safety. So thank you. And just a few words for my colleagues, three colleagues. One, Elisabetta. She will present herself, but she works with me since November, and she started a PhD on the development of Listeria monocytogenes conventional and PCR method for detection. And then Marina Torresi, we will report some experiences in the past, but also a different usage of data coming from next generation sequencing. And at the end, Alexandra will summarize the experiences that we are carrying out regarding new generation, next generation sequencing. So I give the floor to the chairman of the session, and we are here for questions. Maybe we'll give some help to the colleagues if it's needed. And thank you another time. I hope that you will enjoy the presentation, and the presentations could be a start for new collaboration. Thank you, and we will see during the web conference. I don't know if the moderator is available. If not, I'll give the floor to Elisabetta for the first presentation. Go ahead, please, Elisabetta. Thank you.
Hi, good morning, everybody. For my first presentation, I'll share with you my screen. I'm sorry, I cannot share my screen. with my those should allow me to share the screen. Okay, there you are. Thank you. Thank you very much. So, what is it? Okay. Okay, so we should go on presentation mode. Can you, everybody can see my presentation? That's great. Okay, so for the first presentation, today we will talk about state-of-the-art of detection of Listeria monocytogenes from tested samples in a laboratory, food, environmental samples, and diagnostic specimen. So today we are going to talk about new and recent research. What are the newest knowledge, and we'll see what are the most problem. I don't know what's happening. Okay, so we'll talk about what are the newest knowledge today, especially on some Listeria monocytogenes strains detection that are giving us some problems. So we will walk through the detection workflow. We'll try to understand how to do it, how we do the detection of Listeria monocytogenes to try to understand where is the problem and how we can solve it. So first of all, I would like to show you this research. Okay, in this slide, I reported some recent research, some new knowledge, where you can see the red circle, which belongs to lineage 1 Listeria monocytogenes strains. And as you can see, the biggest red circle, Connor complex 1, is they are the strains that are most commonly isolated in clinical cases. On the other side, there are the blue circle, CC9 and CC121, belonging to lineage 2, and those are the strains mostly isolated in food and food environment associated samples. So as you can see, there is a neat difference between lineage 1 strains, lineage 2 strains, and especially those strains that we are isolating from food, like CC1, which is the major cause of disease and clinical cases outbreak in human and animals as well. And the major problem is that this CC1 is not closer to the one that we isolate in the food. So there is this clear difference between strains that we isolate in clinical cases and this in the food. This is the major outcome from this picture that I want to share with you. So on the basis of that, the question that arises is why there is this difference in detecting different strains of Listeria monocytogenes strains. We all are aware that Listeria monocytogenes clinical cases are due to transmission through food. So why CC1 clinical cases are not detected in food and food environment? We'll try to understand this, and we're trying to find a solution, or at least I'll tell you what we are doing, how we are working on it. So in this, I put this slide just because I want to tell you that there are different methods that are used to detect Listeria monocytogenes. The gold standard in Europe is the ISO 11290. But there are other methods, for example, the USDA FSIS. Both of them have some common points, like the enrichment step, first and second enrichment step, that is being done in the Europe with the freezer. In USA, they use a different broth, but the aim is the same. Allow Listeria monocytogenes to grow to detectable levels. After this, there are the plate tracking. You can see that here as well, the two methods have different substrate. They use different plate, but the aim is the same, trying to isolate and find Listeria monocytogenes. So I want to share with you what we do in our lab. I already told you that ISO 11290 is the gold standard for Listeria monocytogenes detection. So it starts with the samples. In our unit, we deal especially with food, so we start from food, but ISO 11290 can be used on a wide range of samples, like clinical specimen, brain, feces, and environmental sample. So we take 25 ml of food of the sample, and we are 225 ml of a freezer. We put them in a stomacher bag, and we mix them together to make it smooth. After this, we put them for incubation at 30 degrees for at least 25 hours. And the next day, we are going to take the first enrichment broth of the day before. We take 0.1 ml and we pass it into a test tube with 10 ml of freezer. At the same time, we take a loop from the first enrichment bag, and we use it to strike it on agar plates that, in case of ISO, is aloha, agar, Listeria, agostini and octavi, and another plate, a second plate, that is the Oxford. We incubate the tube test, the second enrichment broth, for another 25 hours, and the next day we do the same. The plates are then incubated for 48 hours. And at the end, after 48 hours, we are going to examine the colonies, or we are going to examine the plate, and hopefully we find what is shown in the picture. The variation of the agar to black and typical colonies of Listeria monocytogenes on Oxford, and blue-greenish colonies with an halo for colonies grown on aloe agar. After this, all suspected colonies are taken on a nutritive agar, not a selective one. This to allow poor colony growth that we are going to use the next day for more confirmation tests like VTEC or MALDI-TOF. So this is what we do now. As you can see, this detection method is quite long, and especially it introduces a bias. This bias is given by inhibitory agents like antimicrobials moleculars, like lithium chloride and others. That is true, they are used because the plates are selective, and they doesn't allow other bacteria like gram-negative or mold yeast to grow. But it's possible that they might introduce a bias to what it's our aim. So find specific Listeria monocytogenes strains. So going back to what is our talking today and our major question, we are having problem finding some Listeria monogenous strains, and in particular 4B strains and Connacht complex CC1. So why, what is the reason why we cannot find these strains? Could be because of the bias introduced through the detection method. This might be an option. But if that's so, why are we keeping doing the detection method? This is why. In this slide, I show you some pros and cons of conventional methods. First of all, obtaining a poor culture of Listeria monocytogenes allow us to stock the bacteria. In this way, we can have a bank that we are able to use for further tests like antimicrobial resistance or biofilm production tests. Even more, to have a proper identification of Listeria monocytogenes, so to be able to subtyping properly the bacteria that we isolate, we need to have poor culture, especially for tests techniques like whole genome sequencing or next generation sequencing. So yes, the main cons is that it requires a lot of time, it's time consuming, it requires effort, and it requires training people. But conventional methods can be used on a lot of different kind of samples, and we need conventional methods to isolate the bacteria and have poor culture. So, we need to have poor culture and we need to carry on with detection methods. So what could be another problem other than the bias introduced with the detection method? This slide summarizes a bit what is strains diversity. There are different Listeria monocytogenes strains. We can see that there are two major lineage 1 and 2, that we know. Lineage 1 is mainly involved in clinical cases like Ancona complex, like CC1 and CC4 are usually isolated in abortion and neurological disease for humans, or ruminant rhomboencephalitis for in animals. But what I want to really show you with this slide is that these two type of strains, these two different type of Listeria monocytogenes strains present different genes, so different abilities. For example, the strains isolated from clinical cases show genes for resistance to low pH, for gastric environmental. So while strains isolated food environment are usually harboring genes for resistance to disinfectant, antimicrobials. They have an increased ability to form biofilm, and they present stress survival island genes, which makes us think then, and it confirms what was seen in previous papers, that lineage 2 is more fitting for environmental survival. These strains are able to survive stress from inhibitory agents, heat stress, pH stress, all kind of stress linked especially to food and processing food and food processing environment. So the difficulties to find the 4B strains can be because there is this diversity between the strain itself, or because there is a competition between them. So that's our experience. We already saw that with our research, we noticed that in one sampling point, which is represented with the red line, with the blue line, we can find more strains in the same point. So more strains for one sampling point, and more strains can be detected in the same environment. This next slide show even better that more strains, more cluster of Listeria monocytogenes can harbor the same environment. So is there strain diversity, or is there a competition among them? And especially if more strains can harbor the same environment, why more strains cannot be found in a sample? This brings me to this slide, where I want to show you another experience, something else that we notice. If we collect 2 strains from a sample and we subtype those strains 56 times and find one sample, one pulsotypes. But if 15 times, we can find 2 pulsotypes. If we sample and subtype 3 strains from the sample, 160 times we can find 1 pulsotypes, and 40 times we can find 2, 8 times 3, and so on. More strains we subtype, more possibilities to find different pulsotypes, so different Listeria monocytogenes type in the sample. So this brings me to a question that I would like to share with you. How many strains are needed to have a clear scenario of Listeria monocytogenes contamination in a food processing plant? So how many strains, how many colonies we should collect to actually represent the reality of Listeria monocytogenes convention? So could be 1, 10, 100. So we will give you the answer later on, but I would like for you to think about this. It's something to let you think about. So we have seen, so far we have seen possible problem with detection method. We have seen possible strain diversity, and the last thing that I want to talk, that could be another reason why we are having problem isolating and detecting some type of strains, is strain competition, or back to enrichment bias. So strain, our colleagues in Greece show us that if we put 2 strains in the same enrichment broth or the same food, after 24 hours after, when we're going to detect the strains after hours, we are not actually finding both growing at the same rate. So their growth is not equal. One strain can overcompete the other one. And on the basis of that, I would like to show you what's my experience. When I started my PhD, I had to understand how these Listeria monocytogenes strains were growing. So I went on Combase, that is an online tool for prediction of Listeria monocytogenes growth. And as you can see, there is no actually difference between strains. So Combase tells me that all Listeria monocytogenes strains are growing in equal measure and rate. The only difference can be given by external factor like temperature. This is, for example, at 4 degrees. But what I'm learning from my research is that if I test each strain singularly, and I would like to tell you that these strains are all 4Bs, so I used different 4Bs and put them in the enrichment broth freezer. You can see that not all the strains grow at the same rate. There are some that grows more and there are some that grows less. And even more important is that the same strain put in an enrichment broth that is not as selective like the half freezer, but nutrient one like brain heart infusion, these strains actually grow and it can grow as the others. So all these questions that I share with you, all these doubts that I share with you so far, detection bias or strain diversity, strain competition, they are still open question. But from my point of view, this data, which I need to increase anyways, look promising. So from this, we have seen the problem and all the opening questions that that we are having. How can we solve that? We thinking about difficulties in 4B strains detection and thinking that there might be more strains than actually just one in the sample that I'm analyzing. The first bet, my first bet is magnetic beads. So what I'm doing with my PhD research is to introduce magnetic beads in broth enrichment to allow collection of more strain possible in the enrichment, trying to not lose any strains, also the one that might be underrepresented in the environment or those are underrepresented in this case in the enrichment broth. Next will be the real-time PCR or quantitative PCR. So what it's my idea and what I want to do is that using the PCR for real time, I'll be able to have a picture of at least an idea of what is in the enrichment broth to be sure that the strains I'm looking for is there. And with the quantitative PCR, even better, it could tell me how much is there. This will allow me to know how many colonies I need to select to actually find what actually find the strains I'm looking for. Finally, the last bet, probably the most challenging, is metagenomics. This will allow me to study the genomic trait in the enrichment broth to see if the strain, the DNA of the strains I'm looking for is actually in the enrichment broth. It would be even better if it could be done straightforward in the food. But at this point, we are still, it's still early technique for us to implement it in food. So we will see in the next few years. And with this, we just summarize a little take-home message to how solve the 4B mystery, or at least this is my point of view, and these are my top point to remember. And I hope that other people share the same view with me. First thing, more strains can be found in one sample. So we need to understand how many colonies are needed to be collected to actually represent a food processing plant situation of Listeria monocytogenes contamination. There is no correlation one-to-one in a positive sample. So if you have a positive sample, it doesn't mean that there is only one strain, but more strains can harbor the same sampling. And finally, new detection method like quantitative PCR could be a good and screening method to have a better subtyping. So this is some reference. Thank you for your attention. And I would like to pass you now to Dr. Pomilio for some more few words.
Okay, thank you, Elisabetta, for your presentation. Just few comments regarding your presentation. What you need to take from that presentation is that you need to select and subtype more than one colony, because based on the data presented by Elisabetta, around 30% of the food samples or food environment plant samples present more than one clone. And we are not checking how is the growth of the 4Bs, because we think that 4Bs are less perform less during the growth. But the second problem is that what is the ratio between the presence of 4Bs, so the concentration of 4Bs, and the concentration of, example, one or different clonal complexes like 2A in food or other samples. If you have a ratio 100, you need to subtype around 100 colonies. And this is why we are putting in place screening methods to know if in a sample there are more than one clone. So if there is more than one clone, you need to perform selection and subtyping of a lot of colonies, but we can't do more. This kind of approach is now used also for detection of E. coli stack or VOTX producing. So it's the same. And at the moment, we cannot do nothing more because media selecting the different clonal complexes are not available. So thank you for your time. This was the first presentation. I don't know if the moderator needs to take the floor, or we will continue with the second presentation. And because I don't see any hand raised for question, and if you need, so if you have question, please send us a message or raise your hand in the room of the presentation, and we will be happy to answer all those questions. So I give the floor to Marina for the second presentation. Is from the NGS and back. I kindly ask Marina to say something about her. And I give the floor to her.
Good morning.
Thank you, Marina.
Okay. Good morning, everybody. My name is Marina Torresi, and I am a biologist, and I work in the food hygiene unit since 2008, mainly in Listeria typing. So I'm going to share my screen, hoping to have no problem. Do you see my presentation?
Yes, yes, go ahead.
Okay, perfect.
You need to go in presentation.
Okay, is it in presentation mode?
No.
No.
So close, go in presentation, and after you open. Sorry.
Or
Now?
No.
no, if you go on bottom of the screen, right hand side, close to that line to zoom, bottom of the screen.
Where? Okay. Now it works because for me it's the same than before, but for you it's okay? No.
No, Marina, please close the presentation. Go with the file to the presentation mode, and then you share another time. Thank you. Please open the presentation now.
Dove fa? Qua. Qui? 2? Ma ho fatto.
You're talking in Italian.
Yes, I know. Okay, I open presentation. It's
sorry.
like before. The problem is the double screen, I think.
Disconnected. Okay, open, because we are wasting time. And
But can you see now?
no, I see my name on screen. Disconnect the second screen. The second.
Miss Marina, kindly listen to me. Just share your screen and click on the share screen. Yes, now we can see. Now we can see.
Go ahead.
Oh, it works?
Go ahead.
Okay, perfect. Okay, let's go. Okay.
Okay, now,
so,
now press, listen. Now press F5 and then it will play.
I pressed F5.
Okay.
It's okay?
Yes. F5.
No. F5, yes. Are you able to see?
No. F9, sorry, no. F9.
F9.
Yeah.
Okay, I pressed.
Marina,
can you see?
No problem. Go ahead.
Okay, go ahead.
Okay.
Go ahead.
Alt F5.
Can I start?
No, go.
Okay. I will describe in my presentation how we managed an outbreak occurred in Italy 6 years ago. Thanks to new generation technique, in particular to next generation sequencing. Before starting, I would like to highlight some statements. Probably most of you know that there are different methods to type a strain. But in the last years, whole genome sequencing, WGS, offers the highest level of bacterial strain discrimination. So when you perform WGS on a strain, you can know what you want, you can do, and you can study.
You're not still seeing presentation.
You know,
Can you move that one here?
Marina.
Yes.
If I have the last edition of your, I will. Okay, now it's good. Number 3. Okay.
It's good.
Yeah.
Okay, even if it's not in presentation mode, it doesn't matter.
Okay. Okay. It's okay.
Okay. So, okay, I was here. Say that if you perform WGS on a strain, you can do and you can know what you want, and you can study what you want. So NGS and bioinformatic approaches have been used and will be used to in the next future to identify outbreak origins, track transmissions, and investigate epidemic dynamics. Furthermore, in the last until 2 or 3 years ago, WGS was an expensive method, but now the decreasing cost of WGS and computational requirements has made WGS more achievable for public health laboratories. A main goal, a focal point for public health laboratories during an outbreak investigation is to understand which are the strains related to the outbreak and not, because usually when you are performing, when you are in an outbreak investigation, you receive in your lab a big, huge amount of strain, and you have to understand quickly which are the strains related and not, because besides the strains, there are people, there are establishment and people waiting for your answer in order to limit economic loss and obviously in order to preserve consumers' health. So this slide summarizes the contents of my presentation, and I will start with a brief description of the outbreak, then the workflow in the lab, our typing results, including the development of an ad hoc real-time PCR assay. Validation of this assay, and finally conclusion related to advantages and disadvantages of our management and our methods. In the first part of 2015, there was an increase of listeriosis cases in Marche region in central Italy. And so the official lab of that region asked help to understand the situation and to type the strains. We found correlation among some clinical cases, and this was the beginning of a 2 years outbreak involving at the end 24 listeriosis cases, all hospitalized, and 4 people died. During this period, we received in the lab more than 1,400 strains, mainly from food and environment, and 37 clinical strains. In the right side of the slide, you can see which were the analyses that we performed. First of all, species confirmation, and then type, starting from serogroup and serotype, and then going in deep with the PFG, MLST, and SNP analysis. The last two methods were performed with the WGS. As you can see, and as I said, we developed also an ad hoc real-time PCR used as a screening method. Here, there is a summary of our results. most of the strains, both clinical and non-clinical, were serogroup 2A. That is usually the prevalent serogroup in strains from food. And here also in clinical, because the outbreak strains was a 2A strain. As you can see, also regarding PFG results, so pulsotype, we have for clinical strain only one prevalent pulsotype M, that was the pulsotype of the outbreak strain. This pulsotype was also prevalent among the non-clinical strains. Because these results are regarding the strains from traceback and forward activities. So samples were taken in establishment epidemiologically linked to clinical cases. So at the end, these are the genetic characteristics of the outbreak strains. It was a 2A strain, serogroup 2A, having a PFG profile named at SCDC as APA 246 ASC 356. It was isolated in a hoghead cheese sample that was the probable source of this outbreak, obviously together with other pork products, and there was a problem in the establishment. We found the same strains also in the environment of the establishment. Now I want to highlight which was the flow of the strains in our lab. I mean that we didn't receive the strains in a regular, a constant way, but before understanding which was the source of the outbreak, we received a low number of strains. After detection of the strain in the hoghead cheese sample, we received more than 1,000 of strain in 2 months. So we had to work hardly and rapidly to give answer and to the food business operator because there were products detained and people waiting for our typing. So lucky for us, colleagues from National Center for Genomic Sequencing, also here in Teramo, were working on development of a real-time PCR, a screening method starting from whole genome sequence. This slide summarizes what they did. So they started from sequences of both strains related and not to the outbreak. They performed annotation and pangenome analysis with PROCA and RORI software. And then they analyzed the output of these two software in order to find regions common to the outbreak strains but absent in the other. These regions were then aligned with another software, cluster BU, in order to find sections having 100% similarity that can be used as potential target for real-time PCR. These sections were then imported in another software, Primer Express by Applied Biosystem, in order to find the best choice to develop the real-time. Before see the real-time characteristic, I want to show you more in detail what they did, trying to understand the different steps. So this is a neat map. This is a file and output of Rory software. And as you can see here, it's quite simple to understand if there are regions common to your outbreak strain in blue and absent in the other strain in white. So this is a first approach to see if you have some regions of interest or not, because it could happen that you haven't. After that, you can analyze another Rory output file. This is like an Excel file in which you have in the rows the genes and in column samples. So you can understand if you have regions common to the outbreak strains, and you can understand which are the genes involved. Starting from here, from this code in Rory, then you can go to see the same code in another software, Proca. This is a Proca output file, and as you can see, there is the same name, the same code for the gene that you had in Rory. From here, you can understand the exact position of your region, of your genes, because here you have the initial and the final nucleotide in order to cut your whole genome sequence and to import them in cluster view, because with cluster view it's difficult to analyze the whole genome sequence. So you have to cut your sequence and to import the cutted section in cluster view. ClusterView is another software able to align your sequence in order to find the sections having a 100% similarity among your strains belonging related to the outbreak. So these sections are the potential target for the real-time PCR and are the input for the last software, the Primer Express by Applied Biosystem. This software works with the parameters listed here on the left and give you the best choice to for forward, reverse, and probe. As you can see in the last column, there is also a penalty score. So all results are sorted by penalty score. So you can choose which are the section having the minimum penalty score. In our case, we chose 2 regions having the minimum penalty score. And in particular, so we performed a duplex real-time PCR, having as target a recombinase family protein REC and a transcriptional regulator trans. After the choice of the target regions, obviously we had to perform validations. And so we calculated the efficiency, limit of detection, sensitivity, and specificity. We obtained very good value for efficiency, for load, and for sensitivity, and we obtained a specificity of 95.6. This was a good value for us since the method was developed as a screening method. So for us, it was important to avoid the false negative, and it wasn't a problem to have some false positive, later detected with other techniques. In fact, the method real-time screening was tested on more than 800 strains, and at the end we obtained 24 false positive, 15 detected with PFG, and 8 detected with SNP analysis. In fact, as you can see, there were 6 pulsotype different from the epidemic cluster pulsotype, having positive results in real time. So these false positive were detected thanks to PFG. We obtained also some strains having the same pulsotype of the outbreak strains, but thanks to SNP analysis, we understood that that strains were not related to the outbreak. This slide shows the lab workflow without and with the real-time PCR screening. As you can see, thanks to this method, we were able to decrease the laboratory turnaround time from 41 days to 8 days. So it was very important. We were able to decrease time, but also cost of analysis, because we performed the more expensive analysis, in particular PFG and WGS, selected strains. And this was really important because, as I said at the beginning, our main rapid typing to the business operators to have between strains related to and not. So at the end, without the advantage, on the contrary, there are also some disadvantages. Real-time PCR development was quite labor-intensive and not easily standardizable, and it needs high level of expert and skillness in the Listeria biology and bioinformatics systems and platform. Furthermore, 6 years ago, we had another problem. WGS was not standardized and accredited, and so we had to match this technique with other laborious and expensive assay as PFG. But nowadays, things are going better. So there are a lot of laboratories that are going to accredit this method. So this disadvantage is disappearing. Well, thank you everybody for your attention, and thanks to my colleague Alexandra Chiaverini, who is here today with us, and Marco Di Domenico, Massimiliano Orsini, on all the staff from both National Reference Lab here in Teramo.
Thank you, Marina, for your presentation. And I don't see any hand raised for a question. I know that the presentation was not easy to, it's really difficult, but we are available. If you need assistance, and together with the National Reference Center for Genomics, and to apply the same approach also with different softwares. If you are working in the field of foodborne outbreak investigation, remember that we are in the veterinary side, and then if you are investigating a food processing plant in Italy, you close the food processing plant for trade if the processing plant is in foodborne outbreak investigation, and you need to give to them a fastest answer as possible. Cell this was why it was mandatory for us to put in place a really fast method. Also, if the method gave some so too much sensitivity, but it was fit for purpose at that time. So now, to summarize all the experiences that we had and we are having now, and is still open the discussion and the conclusion of the activities, I give the floor to Alexandra Chiaverini. And Alexandra, please present yourself to the participant. And for the next presentation, you have around 20 minutes.
Thank you, Dr. Pomiglio. I am Alexandra Chiaverini. I am veterinaria and I work with the unit of Dr. Pomiglio since last year. And my main activities are to managing the whole genome sequence data for different fields of our laboratory. For my presentation, I would to share which are our current application for whole genome data for different fields of our research or our laboratory. Before doing that, I would like to give you some pills about the history of the sequence or the sequencing platform that was born in 1977 and was historically used for MLA and MLS-TSA, but was this first generation sequencing was replaced because it was very time consuming for the next generation sequencing, and now the third generation sequencing. The real difference among this platform are the output that they give, because now we can allow to obtain a single molecular resolution for DNA in real time. So the time and the cost are very reduced. For the application on NGS platform for the food laboratory, we have different way. We can start directly from the strain isolation, so we can use the single culture, and we can go on with the whole genome sequencing, or otherwise we can use directly the food matrix. For the food matrix, we have two ways. We can go for the shotgun metagenomics that is usually used when we don't know. Yes. When we don't know what are the microbial communities in the food matrix, or otherwise, if we want to perform taxonomic studies, we can go on with the target metagenomics, which the very most famous is the 6ES target metagenomics. Otherwise, we can do figure analysis using the metatranscriptomics. All the output that we obtain from the different techniques of sequencing, we can use the output for perform pangenome, cone genome, for study the genes and the relative function of these microbial communities, for assess the phylogenetic composition, and for study the microbial activities. That is very important, this last part for food industry. We usually use the whole genome sequence data that it's characterized by several steps. Which one, the quality control is the most important, because obtain a very good quality data allow to us to perform a very good quality analysis, and then we can share the information between laboratories. For this, we usually use the quality control threshold guideline recommended by the GeneTrack group that in their study they obtain which are the quality threshold recommended for several foodborne pathogens. Here, for example, you can see for Listeria, which are the parameters to follow for obtaining the good quality data. But which are the current applications for bacteria foodborne pathogens? We usually perform the comparative genomics analysis for assess which are the relativeness that occurs among isolate. This was very important because it's constituted by two main activities and analysis: core genome LST and the SNP analysis. They are very important because they are very most discriminating instead the MLST sequence type and the canonical SNP that was frequently used for Bacillus anthracis. And the SNP analysis and the core genome LST allow to us to perform outbreak investigation, to perform the lineage-specific analysis. It's very suitable for the global surveillance. In this context, what is essentially the core genome LST analysis? This is the next step of the MLST because it relies in the detection of the alleles consulting the Pasteur scheme that was constituted by 1,748 genes. And the sequence and the order of the gene that we obtain by this analysis can be used for perform the cluster analysis and phylogenetic. When I reported one example, I reported an output of the core genome LST analysis that is represented by the minimum spanning tree, which is the graphical representation among the relatedness among the isolate. Is a tree constituted by node that is represented by the strain, and the branch that show the distance that occur between isolate. This was a very interesting study that we done this year because we investigate the Listeria monocytogenes strain that are circulating in two different food processing plants on central Italy. And the cluster analysis together with the virulence gene detection and persistent gene allow to us to detect which are the persistent gene, the persistent strain. The same work was performed also for salmon facilities industry, and where we studied 19 persistent strain of Listeria monocytogenes, also with the help of the characterization using detection of the virulence gene and the persistent strain. The core genome LST analysis, it's also used from Pathogen Watch. There is a global platform for genomic surveillance. Is suitable for different pathogens like Klebsiella pneumonia. In this output, I reported the relativeness about our isolate of Klebsiella pneumonia strain isolate from wild animals, which all the available clinical isolate from the worldwide. Here you can see that our isolate is very similar with other 138 genomes that are isolate from different countries and other world. Furthermore, in the comparative genomics, we have the SNP analysis. SNP analysis relies in detection of the single nucleotide polymorphism and which occur along the genome of the microbial organism. And the detection of this SNP can allow to establish the phylogenetic study. The SNP analysis was also important for assess the foodborne outbreak investigation that was previously presented by my colleague Marina. And the SNP analysis, as previously mentioned by Marina, allowed to us to identify 2 different clusters that occur in same time during this severe outbreak of listeriosis in central Italy, and allow also to identify the source of the infection. After that, our team published also 2 genome announcements about this outbreak, because one genome announcement was related for the whole genome characterization of the 2 strains responsible for the outbreak, and then was published the re-emerging Listeria isolate that occurs 3 years after the outbreak. For the SNP analysis, we have another tool that is a free available platform that is pathogen detection. We usually use this platform for verify in real time there are SNP clusters that occur for Listeria monocytogenes. But as you can see, this platform is available for different pathogens as Salmonella, E. coli, and Campylobacter jejuni, and so on. What gives this platform, as I previously said, is to give information about which are the SNP clusters that are currently gone, and can give you the metadata regarding the isolate. So you can see which are the hosts, are the country that submitted the sequence, and when was submitted the sequence. So it's global in surveillance. Furthermore, we used the phylogenetic analysis also for not only for food pathogen bacteria, but also for foodborne pathogen viruses, as hepatitis E virus. This virus is a real concern for us, especially for our region, for public health. And what we did is to cluster all the sequence available from NCBI of hepatitis E virus, which with some isolate that we identify in wild boars. And what we obtain is this clustering analysis using a different method statistic that is called maximum likelihood. So what we see is the similarity about the strain. And what we see is first, which is the genotype that are circulating in Italy, in our region in this period. And then we detect 2 different genotypes that are very related with the classical genotype of pig. So it's showing some interconnection from wild boar and pigs. The SNP analysis, furthermore, was performed also for the genetic characterization of Klebsiella pneumonia from wild animals. That it's a work that I'm doing now. We have collected several samples during 2 years from wild animals, like deer, red deer, magpie, European badger. And we obtain 17 positive isolate. We have sequenced this isolate. We obtain this cluster analysis where we can see that the cluster analysis is accorded with the sequence type result. For the last, Marina, my colleague, presented what we did for the pangenome for establish all the real-time PCR assay. But furthermore, we are using the pangenome deriving from the outbreak strain of Listeria monocytogenes for performing the genome-wide association study. That means that we are looking for the high significant gene that are associated with the re-emerging of this outbreak that, as I previously mentioned, occurs free after the outbreak. This pangenome analysis, this is the output, and it was performed in according to the SNP analysis and in according for the core genome LST analysis. The pangenome was used for Listeria monocytogenes, also for the past, because allowed to report the first finding of Listeria monocytogenes sequence type 128 in a dolphin brain. This was very particular as finding because usually this sequence type is associated with food. Then another research field that we are carrying on is the antimicrobial resistance for Listeria monocytogenes, especially in clinical isolates. We have work that was accepted for the European Public Health Conference for the year. And what we do is the epidemiological study and characterization of more than 233 isolates collected from clinical specimens during 8 years. Furthermore, we also submitted this work about the genome announcement of Serratia rubidae isolate that was very particular because usually Serratia rubidae is usually isolated from environment of food or from plant, but we found this particular strain in a food product that show particular antimicrobial resistance profile and harbored to the different bacteriophages. For the last but not the least, this is very important for me, it's the LEGO project that is funding from the Erasmus European Foundation. This project where the Dr. Pomiglio is the scientific manager, is focused on to perform this e-learning course for everyone, for build a new figure for the research activities, the food microbial bioinformatician. This project was in collaboration with 3 European partners, as Helsinki University, as Bignosh University, and IENIA from Spain. That was very important for me because I was involved in the coordination of the e-learning course, where the student can approach to all the whole genome sequence world and for all the bioinformatician analysis. Thank you for your attention.
Oh, thank you, Alexandra, for your presentation. This was a slideshow presenting some of the activities that we are doing in the field of genomics mainly. And this was the last presentation of the day for us. So now what we can do. We are in Italy, and yesterday we had a good discussion with the colleagues of the university and with the organizer of the meeting. So now what we can do together, the distance is not an hurdle. Until now, we can work at the distance and building a new collaboration, building studies, increasing the competencies and exchanges mainly. So we do not have until now questions, but the organization will send to all of you the addresses, or maybe you send to them questions and they will forward to us as you want. What I expect from today is to start a collaboration with, I can't see all of you, but maybe for some for sure, but we can build some new projects of collaboration. Alam Khan has the hand raised, so I give the floor to him for maybe a question. And we wait for questions, and we are open for collaboration. So if you are interested in the framework of OIE to build competencies together, of course, working, giving the different contribution from the university, from the national institution, from laboratories, we are open to collaboration. And before I give the floor to the moderator, to the chairman of the day, I don't know if I saw before a hand raised or not. I don't know if I give the floor to the chairman of the session, saying to all of you bye-bye. And if you want, and if you consider our collaboration good, we are available to build projects and to build activities with all of you in the framework of OIE, in the framework of European Union collaboration, and also in the framework of the National Reference Laboratory for Listeria, but also, as you realized before, for Klebsiella and for viruses, or for other bacteria, because in our Instituto, and I kindly ask you to check the website of Instituto to find the possibility of collaboration with us. So I saw now Tariq is on the video, so I'll give to you. Sorry, my camera was off. I give the floor to you for the last words, and we are here for next and for more day of information and to collaborate together.
Professor, gracias.
For you.
Thank you very much. Thank you very much for your final words. And now, dear colleagues, the session is open for the questions, and all the respected 4 speakers are available to face your challenging questions. Please, if there's any, we would request and expect the questions from the listeners. It seems that I would like to ask Professor Francisco and his team a question. Professor, I have a question. Dr. Jamal is with you. In addition to moderator, I got a question. The question is that as you see, this global warming is becoming more and more reality. It was a reality, but now it is becoming a hard and tough reality. So in the wake of global warming, when the world is turning toward the food preservation, the cold storage, the deep refrigerator, and seeing the capability, the efficiency of this bacteria to grow on the low temperatures, as I read in some literature, that this bacteria has the capability to grow on the low temperature. So what you and the other members of your team think that the Italian researchers or the Pakistani researchers would do to stop this challenge when this bacteria is capable to grow on the low temperatures? How are we going to cope this problem? This is my question.
Yeah, okay. Thank you for your question. So remember 3 years ago, the outbreak that we had on frozen corn. And we know that Listeria is able to survive and to grow also at the refrigeration temperature. And Mohammed, please, could you switch off the microphone? Okay. So when you go into the thawing of the food, you need to rewarm the food and to go at least into the pasteurization temperature. Listeria is not a good microorganism, and it is able to grow also at 4 degrees, but to survive for a long time at -20 or less temperature. So what we need to do if we have a frozen food, it's better to reheat the food, except if it is ice cream. But we know that also ice cream could be a risky food for Listeria, and we need to take all the measures to limit that growing, the proliferation of the bacteria. We know that Listeria is not really common, and the number of cases around the world is not so high as for Salmonella or Campi or maybe viruses. But we know that from 20 to 30% of the sick people, of the patients, will die, at least in Europe. So the disease is a severe disease, and it's really expensive because in Europe, based on the burden of the disease, we calculated around €1 million the cost for one clinical case. So we need to take into consideration the ecology of Listeria, to work hard in the food processing plant, but also at our place, because recent studies of source attribution done by French authority called ANSES calculated that the rate caused by listeriosis for bad storage of the food at home is able to give 1/3 of the cases. So it's important to put in place surveillance in food processing plant during transport at the supermarket, but also in the refrigerator we have home to store the food at the right condition and the right temperature. So, Tariq, what you...
Yes,
is it okay for you?
Answer addressed the question in some ways, and thank you very much for giving this answer. Professor, with this, I would like, with your permission, to move toward the final stages of today's gathering. And before I announce the end, I would like personally name and say special thanks to some people who really worked hard on making this gathering possible. I will start with your team, Professor Francisco Pomilio, and the other three members. They were very good, Miss Alexandra, Maria, and Marina, and we would love to see you and your team member here in our university in the future once this corona is past. And here a lot of faculty members, they are enthusiastic and they love to work with you, and we welcome to see the wonders of the Cholistan Desert and our camel gift. So, and thanking from your side, I would like to say thanks for some people on our side who really worked very hard. I will start with our patron-in-chief, our Vice Chancellor, Professor Muhammad Sajjad Khan, for giving us the permission for this all scientific endeavor. And then I would say special thanks to Professor Mazhar Saab for realizing this gathering, and Dr. Sabika for her hard work, and Dr. Abdullah Saghir for participating in this gathering. And special thanks to Director IT for handling the technical issues today, and to my respected attendees and the faculty member, thanks for your patience, time, and energy for listening to this seminar. And these are my final words as a moderator, and I would like to invite Professor Mazhar to say a few words. Professor, you're welcome to talk to our respected guests, and that's how we're gonna end toward our meeting.
Okay, I would extend all of my wishes, good wishes to all of you and including your team. And I would also extend that our institute or our university would welcome you all people here. And we will also extend our cooperation in near future also. And if you get any opportunity to have a collaborative work with us at CUAS, we will also welcome you, because OIE is considering many other regions, but this is our neglected area. So I would also request you people to please consider us also in your schemes as well. And that was a great opportunity to have a good interaction with you and your entire team, and also your unit. It was very beautiful presentation that all the presenters made, and all of our participants might be having their some food for thoughts. It is timely to say here that 300, more than 300 or approximate 360 people registered this e-conference, and the participants were from more than 7 countries, including Malaysia, Hong Kong, and Argentina, and Egypt, China, Saudi Arabia, and many more countries also, and Iran, Turkey, exclusively. So I would also thank to the participants, our scholars. There were veterinarians, and there were academicians, and there were also scientists who participated in our 2 days seminar. I would also extend my thanks to my faculty who had a patience for to learn from you, and also to listen your speakers also. So that's all from my side. And I would also request Professor Francisco that we will extend our cooperation even after this e-conference also. That's our, and I'm also thankful. Yes.
No, please go. Sorry. Go. I have just 2 words to say.
Yes, I'm also thankful. Yes, I'm also thankful to my team also, including Dr. Jamal, who moderated this event very beautifully, and our the backstage team members who performed well. Thank you. I over to the Mr. Jamal.
Professor,
just few words. Sorry, I wait for an email, because we said yesterday to plan a meeting with you next week, and based on your availability, you send me an email, and I'll try to. I will be with you for a talk regarding collaboration. Okay. For next week.
Professor, this meeting, this gathering was the tip of the iceberg, and a lot of scientific emails and MOUs will be happening between us, and science is the reason we are all together. And I assure you on the behalf of all my faculty, much more gathering will be there. Thank you very much. And at last but not least, I would say very much thanks to our colleague, Dr. Tariq. He was the key soul for arranging all this gathering. We forgot to say thank you in the start. Professor Familio, once again, and Dr. Marina, Maria, and Dr. Alexandra. Thank you very much. Bye-bye.
Bye-bye. Bye-bye. See you soon. Bye-bye. Have a nice day, afternoon for you, for us. Bye-bye.
Bye.
Bye-bye.